how is it that a multinational company launches, say, an omeprazole at 24 rupees, and how is it that Dr. Reddy's launches that same omeprazole at 2 rupees? Coming as I do from a multinational company earlier, at AstraZeneca, I remember the efforts that we had put in to do a phase 1, forget the preclinical, a phase 1, phase 2 and a phase 3 clinical research, almost at that point in time spending about 500 to 600 million US dollars. I remember submitting this document as a registration document to the Drugs Controller of India. As soon as I submitted, the same evening, a photocopy of the confidential document that I had submitted was available with Dr. Reddy's lab. At that point in time, we never enjoyed what's called product patent. Therefore, what really happened was Reddy's lab went ahead, bought the compound from Italy, did what is called reverse engineering. If Astra had taken 10 steps to manufacture omeprazole, they took 11 steps to manufacture omeprazole. They were asked by the DCGI to do a 12-patient bioequivalence study, which was for them to show that biologically both omeprazole of Astra as well as that of DRL were similar and that blood concentrations were same. Therefore, it was suspected that the therapeutic equivalence will also be the same. They were permitted to launch the drug earlier than Astra was. They were capable of launching it at 2 rupees, basically because all they had done was a 12-patient bioequivalence study, whereas a multinational company like Astra had spent billions of dollars or millions of dollars to go ahead and do that work. So, the logic there is, if you have spent enough money trying to launch this compound into, say, the world, would you not want to make some returns out of it? While multinational companies, I mean, this is a point, strong point that the ethics committee people should really look at. While multinational companies have spent billions of dollars trying to get a product to the market, since we in India were not signatories to the Product Patent Act at that point in time, we were only process patents. Therefore, we said instead of a 10-step process, we will have a 11-step process, which will give us a drug faster into the market. Obviously, their prices could be cheaper. Till recently, and for that matter, even the remaining part of the generic drugs industry, what would happen is products that go out of patent can still be manufactured as generics within this country at a much cheaper price, and therefore, you will find that striking difference between what multinationals price their products and what Indian companies price their products for. So, I suppose that's an answer to your question, madam.
Ultimately, when we talk of ethics,
absolutely,
we are talking of the patients.
Absolutely.
So, a patient is bound to prefer a drug which is cheaper than the one.
Fully agree.
Now, as far as the vigilance is concerned, it is taking care of the efficacy and the all the other things. But what about the cost?
I fully subscribe to your thought. Affordability is still a major issue. The government of India has an NPPA, the Drug Pricing Authority, which looks at pricing of essential products. And if you look at the millions of products we have, some of our products are put into the essential product list, of which the government of the country decides on a pricing. The rest of the products, the other products that come outside the criteria of essential products, can have their own pricing strategy as long as the products are patented. If it runs out of patent and falls into the generic category, at that point in time, Indian companies do have a chance just to do the bioequivalence studies and get the drug at much cheaper price. So, that's where the affordability issue gets answered. I knew my first talk on drug development lacked color, so I thought I will have some colorful slides for the second one. This, I think, is the millennium effect. All right, if one looks at 1 for a man, 2 for a horse, when you start developing new drugs and look at, can you have human drugs and veterinary drugs? Human dose will be smaller, veterinary doses would be higher. Is this true is a question that I want to pose before you. Does one size fit all? Unfortunately, one size does not fit all, and therefore, what really happens is, look at a drug company that launches a product, let's say for diabetes and hypercholesteremia, you have to have an adult dose, you have to have a pediatric dose, Mind you, hypercholesteremia at 15, 16. Why 15, 16 at 8 and 12? So, you will have to start working on different dosage formulations of the drug to ensure that you have the necessary doses for pediatric age group, you have one for adult age group, or for that matter, you have to have one for the geriatric age group. We used to have a beta-log metoprolol, which was meant for pediatric age group. We had one for 25 milligrams. We had a 12.5 milligrams for pediatrics, 25 for the geriatric, and you had a 50 milligram for the adult. Which means you have to repeat your studies to ensure that you have necessary data to back whatever you have been doing. Look at the perception of people. 74% of consumers underestimated the average financial investment required to research and develop a new drug by more than 50%, which means today if you are spending $1 billion, people say, oh, they only spend about 500 million US. 45% of consumers thought that the pharmaceutical companies chose to develop only the Me Too and lifestyle drugs with the greatest sales potential, while 71% of those involved with the industry thought that they place healthcare needs first. And 90% of consumers and those involved with industry do not think that direct to consumer advertising provides complete and useful information, while 40% of pharmaceutical executives thought it does. So, there is a lot of money getting into drug clinical research, and therefore, one has to clearly understand that pricing also will depend on the fact that drugs do take a lot of money to get across to the market. Not that the industry is not looking at societal needs. Societal needs today, like I mentioned in the morning, are to look at an Alzheimer's disease, central nervous system, to look at oncology, cancer, to look at diabetes, which seems to be troubling the entire world, more so us here in India, when we have, like I mentioned to you, approximately 12.5% of the population who would have diabetes at one point in time or the other, and you have a variety of other diseases, stroke. Have we been able to develop drugs at age? So, a lot of new drug development is looking at ensuring that we meet societal needs. And like I mentioned again, a variety of compounds get in there, and as you end up with the funnel, you end up with one product that's finally approved. Drug development goes through a variety of phases. Ultimately, for us to ensure that you have a marketable product, it takes you as long as 12 to 14 years, and you enjoy patent for only 3-4 years or 5 years maximum, at which time you need to recoup part of your investment. This is the American public perception about new drug development, and these are studies on mammalians, and frankly I don't like the way things are going. This is what the animals seem to be telling us, and if you look at what animals are saying that way, people want drugs safe and fast, and here is our friend the rat that got tested first, and he says, I go home today, they cured me using this new miracle drug. I'm afraid it will be years before it's approved for you humans. So the rats are making fun of us. This is even more scarier. This is what I'm worried about, and this is what some of my physician friends, including me, must have done in the drug industry. You are healthy, but I have to give you a prescription for Toxicale, says the physician. The drug company rep is totally hot, and she said she would take me to lunch if I sell enough of this stuff, and therefore, will there be any side effects, doctor? The doctor is telling the patient, depends on what you eat. So, some of our drugs, and I'm sure if the rights, safety and well-being of the patients have to be protected, as ethics committee members, you will have to play a crucial role here. If you look at worldwide pharmaceutical development, this is really what's been happening till 2005. Okay, the United States there, rest of the world, you look at European Union and Japan into drug development. Now, if drug development is a new science to drive growth, what really has been happening in the recent past is we are trying to adapt to drug discovery by looking at our success in combinatorial chemistry. You know a product rosiglitazone has a benefit. You combine that with a metformin which also has benefit. Today you are putting together the skills you have of a rosiglitazone separate, a metformin separate, and trying to see if as a combination they could be beneficial. In fact, I'm surprised that the United States is now talking in terms of one drug plus another drug. So they are talking of combination drug effect, and our success in combinatorial chemistry ensuring that. Nanotechnology, proteomics, genomics, and tissue engineering perhaps will be what will run the drug development industry in the near future. And if one is talking in terms of, like I told you, public perception, here is what it is in terms of what the public has been saying from the time 2000 to 2004. If we are holding ourselves back, what really is holding us back in the drug development industry is empirical decisions versus mechanistic approaches, tendency to shoot for the low hanging fruit, the easy targets, and therefore you might feel that we are getting into a lot of me too drugs in the industry. Costs are through the roof despite mergers and acquisitions. I mean, if an Astra were to merge with Geneca, we thought, okay, the combined effort of these two companies will bring down the cost of drugs that they are being researched, but obviously has not happened. We did talk in terms of how the FDA looks at the review process, the NDA drug development review process. Won't spend too much time out there. Here, if one is talking in terms of traditional drug development versus the newer proteomics, genomics, and whatever based stuff, earlier we were empirical, we had traditional approaches which though were rigorous, focused, and ensured statistically valid results were obtained from large populations. But clinicians still treat patients. They have inflexible situations where a couple of our new diseases are defeating the existing form of therapy, and of course it limits our creativity. Therefore, what one has been looking at is application of new science to drug development process, and among examples are simulation models, biomarkers, surrogate clinical markers, and new trial designs. So that's where I think we are progressing in the area of new drug development. In answer to that question that you had, here is the FDA critical path initiative where one is looking at discovery there, go ahead, do clinical development, and after you have done clinical development, evaluate what you have done, go back this cycle and ensure that the goal is to modernize and streamline the scientific process leading to development of new drugs, biologicals, and medical devices. So we are in the drug industry taking a lot of lessons from what has happened in the past to sort of bridge the past to the future. Among the critical path to new medical products, well, basic research, drug discovery, preclinical, clinical, and FDA filing approval process. And if I were to say how the FDA also has been forward looking, I could give you an example of what I did the last year and a half. We were looking at, well, today's traditional medical therapy seems to have failed when somebody has a spinal cord disconnect. He lies paralyzed without recourse to any form of therapy. Could I start looking at seeing if stem cells would benefit them? And the way the US FDA, for that matter, world over, drug authorities are looking at is there an alternate? And if there is an alternate that can translate very fast into clinical use, can we go ahead and use it? I now know of a situation where Osiris, a company in the United States, has approval for conducting a phase 3 clinical research in the use of what's called mesenchymal stem cells in spinal cord injury. How would we like a patient who has had a heart attack, who has an ejection fraction of 26%, which means what? He can't climb those steps of stairs without discomfort, without being breathless. Would it be okay if you look at newer drug development, inject mesenchymal stem cells into him intravenously, those stem cells get there, increase the ejection fraction from 26 to 45 and help improve the patient's condition, which would mean if you still had a lecture here, he could climb steps and get across to this lecture. How would you like somebody who has leg ischemia, inability to walk, a chronic smoker with Berger's disease, inability to walk, hardly can take a few paces, all right? And if this guy, with all of our traditional medicines failing, you go ahead, inject the mesenchymal stem cells into his leg, and lo behold, 2 days after, he is able to walk 200 meters. He's pain-free, he is analgesic-free, and to top it all, in 6 to 9 months' time, if you observe fresh blood vessels being formed, that I think are critical steps that we are taking in new drug development process. So, if the FDA critical path is looked at, they are looking at assessment of safety and predicting it earlier. And therefore, if I say, if I want to predict cardiotoxicity earlier, looking at the Vioxx example, would it have been possible to start looking at using cardiomyocyte cells, injecting Vioxx, finding out that those cardiomyocytes are shrinking? We need not have gone through the $800 million worth of phase 2, phase 3 and a post-marketing surveillance study to detect that people who were taking Vioxx had a QT segment abnormality, which would mean you can predict earlier. Can you prove efficacy earlier? Which would again mean, can you take those stem cells, inject, let's say, a hepatoprotective agent directly to, not the liver though, but the hepatocytes, and see the efficacy of the compound faster, and therefore rush through the process of drug development. And of course, produce all of these compounds at high quality consistently. If one is to look at the shape of things to come, FDA critical path programs out there, here, the next generation therapies and NIH clinical and translational science initiative, they might look like they're all working frictionless, that there is a lot of lubricant out there and ensuring each one moves thoroughly well. But then there are cogs, and I'm sure somewhere up there you have small little bickerings, a little bit of friction, which I think has to be solved if one is talking in terms of seamless drug development for use in the future. We have terminologies that baffle us now. We have proteomics, which is for any given species, the space of possible biomolecules and their organization into pathways and processes is large but finite. In theory, therefore, the biological systems operating in a species can be described comprehensively if a sufficient density of observations on all the elements that contribute the system can be obtained. So, is it likely for us to look at proteomics as an initial goal to rapidly identify all proteins expressed by cell or tissue, which is still a goal that is yet to be achieved in any species? We have known that the genomic sequence between rats, cats, monkeys, and for that matter humans are almost the same. 99% similarity between maybe chimpanzee and humans, and only one rare individual in this entire world who has 10% similarity between the chimpanzee gene and the human gene, and I'm sure you would know who that is. There are more molecular genetic ways to study proteins and more biochemical ways, and what really has been happening is we've been looking at some of this. Well, I wouldn't want to get across and confuse you into what this has been happening. We've been trying to identify proteins and check if by having our advantage, having our innate intelligence in proteomics can help us design drugs faster, that will be more useful in humans. We've been looking at high throughput tests of function. Once you know the protein sequence, for us it's easy for us to say, can I have a rapid high throughput sequence and develop tests faster? We have microscopic localization. I'll check on membrane as well over a period of time, which can again help drug development process. We now can start looking at genomes, we can start looking at genomic DNA there and look at the functional protein, the protein products, how the messenger RNA carries all of this, and design drugs that can work on biological systems faster. I think that's one of the developments that is taking place. You can now have process for protein isolation, and once you are able to isolate the proteins, you can look at identifying them, having measures that will ensure that their quality is maintained, you can digest the proteins, separate them, use a mass spec, ensure you have understood all about the proteins that can go ultimately and get across being a drug and modify the protein abnormality that's happening in us. 2D gel electrophoresis, again I won't spend time on the technique here. These are antibody arrays which again are helping out in a variety of vaccine drug development. These are array based protein interaction detection, again essentially helping in the new drug development process by ensuring that you can get a faster, the faster turnover rate, and therefore ensure time or speed to drug development and discovery. Some of the protein microarray systems that have been useful in designing protein based drugs, again won't spend too much time. Technology of proteomics is not as mature as genomics owing to the lack of amplification schemes akin to PCR. So, we are still dabbling in proteomics, looking at how our initial knowledge of protein base, how initial knowledge of these proteins can ultimately be used to shorten the process of drug development. We now have a new term, and India is pretty good with biotechnology and information technology. You take the T out, you end up with bioinformatics. So, if you have taken out the T there and make it bioinformatics, it's helping us out in development, drug development and diagnostics. If bringing of a new drug is again what it is, this is as per the Tufts. Here is what Walter Gilbert has to say. The new paradigm now emerging is that all genes will be known in the sense of being resident in databases, available electronically, and that the starting point of a biological investigation will be theoretical. While we did talk in terms of how we would have our drug development based on diseases there, if I and you can understand the genetic basis of all disease, would it be faster for us to look at a drug development, say for a breast cancer, when you know you have a Herceptin positive tumor, or you have a ER or a PR positive tumor, would it help us get across to a situation where you can hasten drug delivery? I thought Nitya did talk in terms of how if you knew that somebody, why somebody, talk of Amitabh Bachchan. If Amitabh Bachchan develops, he already has myasthenia gravis, if I am not mistaken. You know the defective gene there. Can you go ahead and design a drug that will ensure that it will still act in the presence of that defective gene, or can you splice the gene out and ensure when Amitabh Bachchan looks at Rekha in the eye, he is not looking at her feet, but looking at her eye and saying, I love you or whatever. Among the rational approach to drug discovery, well, you can identify the target once you have the proteomic sequence or the genomic sequence known, clone the gene encoding target, express the target in a recombinant form, and India of course has taken a lead. We now have Kiran Majumdar's insulin out, we have a variety of recombinant drugs out, we seem to be leading out there. So, we are capable of screening those recombinant targets with available inhibitors, identifying lead compounds much faster based on the biotechnology background we have, and look at synthesizing, modifying these lead compounds, identify fresh ones, look at the toxico-pharmacokinetic studies and have preclinical trials faster. So, our advances in say proteomics, in biotechnology, in genomic technology has helped us do some of this. If we can now start looking at an ideal target, which is generally an enzyme or a receptor in a pathway, and its inhibition leading to either killing a pathogenic organism or to modify some aspects of the metabolism. So, tomorrow we can get across a drug that will identify the malarial parasite based on the genome, and then start looking at would we have ways to detect the disease earlier and effectively prevent people from developing such problems. Now, if one says how bioinformatics can help in target identification, one would look at trying to identify homologous and orthologous genes, the gene order, the gene clusters, molecular pathways and the gene ontology, and then start looking at identification of these unique genes of parasite as a potential drug target and start creating drugs that could cater to it. Here is an example: genome comparison of malarial parasites of human, genomic comparison of malarial parasites of human and rodent, and start looking at comparing the genomes and looking at human malarial parasite and the mosquito genomic sequence, design a drug in such a way that it will tackle all of those mosquitoes which probably have a similar genomic code that can specifically be used to target them. What one should look for, well, this is what the human thing is. There is the genomic sequence, out here is the Plasmodium falciparum and the mosquito, and how proteins that are shared by some of this can easily tackle. An example, again I won't go into too many details of this. Now we are looking at structural genomics and saying can organize all known proteins to families. You can determine structure of at least one member of every family, solve structures of more than 10,000 proteins in next 10 years, generate knowledge and rules from known protein structures, and apply this knowledge to predict the structure of each and every protein of known organisms. And once you know that, you are in a better position to design drugs faster. This is how structural genomics has been able to impact on drug discovery. Again, I won't go into too many details, but you can identify target, validate it, get across to lead discovery, optimization, clinical candidate selection faster, do clinical development, and get across to new chemical entities. A copy or a version of structure based drug design, again for want of time I won't go into these details. A situation where with our knowledge of genomics, with our knowledge of proteomics, we would be in a better position to start looking at a site, looking at a lead, and then optimize that, ensure that you have a drug, having identified the target, ensure you have a drug that specifically targets this area, and you have cell kill or for that matter cure. If one is finally talking in terms of cancer cell growth, look at cancer cell growth appearing to be related to evolutionary development of plump fruits and vegetables. Large tomatoes can evolve from wild blueberry sized tomatoes. The genetic mechanism responsible for this is similar to the one that proliferates cancer cells in mammalians. This is a case where we found a connection between agricultural research, how plants make edible fruit, and how humans become susceptible to cancer. A connection which people earlier thought never existed, they say exists now. This comes out from Cornell University. And if this is true, look at what happens there. There is a small little tomato there, there is the huge one, thanks to whatever agri-bioscience advances we have. Single gene, the ORFX, that's responsible for the QTL, quantitative trait locus out here, has a sequence and structural similarity to the human oncogene whatever. And since we now know that they are structurally interrelated, fruit size alterations imparted by the alleles are most likely due to be due to changes in regulation rather than in sequence structure of a protein. And if I and you decide that we have made major advances in agricultural biotechnology, meddle with those fruit sizes there, a small little tomato to suddenly grow into a huge tomato, eat all of that up, no wonder you have more colorectal cancers in the West. And people are still dabbling on how with all of this meddling of genetic sequencing of a tomato to a genetic sequence of human being, one is prone to sickness. With this knowledge, hopefully we will be able to tackle, say, colorectal tumors much better, breast cancers much better. So the challenges in the post-genomic era, unlocking secrets of quantitative variation. So you can identify the genes FW2, which is a critical first step towards attaining this understanding. We have now knowledge of what happens with colorectals, we now have knowledge with what happens with breast cancer, with lung cancer, prostate cancer, and therefore we are in a position to design drugs faster. Among the challenges, we have genome database, have individual genes with relatively limited functional annotation. Molecular reactions need to be placed in the context of higher level of cellular functions, and therefore clearly understand some of this. We now have in year 2007 a variety of things that are baffling us, but then we seem to be talking in terms of how genomics has helped us, gene identification and characterization. Transcriptomics is expression profiles of a messenger RNA, and how by our understanding of that we are going to be designing drugs faster. We did talk in terms of the functions and interactions of proteins, and how by meddling with that we could have newer drug development. We have structural genomics, we have cellinomics, which is trying to understand the metabolic pathways the cell-to-cell interaction, and of course, pharmacogenomics, where genome-based drug design is possible. In fact, I was asked this question. I said genome-based drug design could one day end up with a personalized medicine. So the question was, which means I can have a drug on my own. A day might come when you will say, all right, all of those with a metabolic abnormality, say genomic based hormonal problem or a metabolic enzyme derived problem, can be tackled by drugs which are going to specifically act against them. With our knowledge of genome, would it be possible for us to say, can I clearly select patients who have MDR, a multiple drug resistant tuberculosis, through a PCR based study, identify only those of them and put them through a clinical trial process, ensure you get a drug faster? Would we be able to do that for AIDS is perhaps the next question. Data mining again helps in clinical research, for that matter in drug development work. Won't again spend too much time. Structural resolution, what structural resolution are organisms different? There is certain amount of relevance out here. Again, I'll just get you across to the next slide, show that drug development looks like that easy thing there, like that small little bicycle. But with whatever is happening in terms of proteomics, genomics, RNA, and variety of newer things, what really is happening is it's not as easy as assembling that bike there, that cycle there, sitting on it and taking a ride. Obviously, you will have to start looking at a variety of parts which are responsible ultimately to ensure that you have a cycle there, and how flexible and adaptable you can make it to finally sit down on that bike. And I am sure at the end of my talk you would say, how I wish I can sit on that cycle and get away from this boring lecture. Thank you so much. I must honestly confess that this part of the lecture was essentially meant more for postgraduates in clinical pharmacology, medical doctors and whatever. But since you are ethics committee members, when you have a situation where you are exposed to say clinical research or a drug development process where proteomics is involved, I would want for us to say, okay, so this is where the level of research is going in, a certain amount of knowledge. If somebody is talking of genomics and saying we're going to design a drug that will probably work for multiple drug resistant tuberculosis, I mean, I just thought you should know, yes, it's possible. And with proteomics now, there is every likelihood you would have protein based vaccines which would be clinical, which would be get across to clinical research as a part of the drug development process. And so this exposure, I didn't get across to some of those slides, but interestingly, there has been question now, and since I have been dabbling in stem cells, the question is, how is it that you inject stem cells intravenously and then they will somehow go find out with their eyes open, how is it that they get across to the myocardium, sit there, improve the ejection fraction. Nanotechnology based solutions where you can tag the stem cells, look at where they are getting across in the myocardium and start looking at the advances of both nanotechnology as well as stem cells. We have been using some of those stem cells to go ensure that you are able to dispense these stem cells out with them, and nano-based technology will ensure that they go and spread out either in the spinal cord for spinal cord injury, in the brain for strokes, or for that matter heart for myocardial cells. Again, for want of time I didn't go into too much of the technology issue. Well, in the first place I don't know technology, and if you try and simplify technology you end up with a lot of problems.
In terms of the new biomed technologies that are going to come in, I think there would be a whole host of ethical issues from a patent perspective, and I think we should have a patent ethics committee.
Fully subscribe to your view, and I'm sure, yes, I mean, if you have a patent attorney or somebody who knows all about patents in your ethics committee group, it obviously will benefit. It obviously will benefit. I will take it across to the industry and say, yes, it is worthwhile, it's worthwhile doing that. But to be very honest, ethical issues in genomic research, DNA based research is still, I would sort of, very, very fluid. And therefore, we might have to get across, sit down together and request Dr. Nandini and Dr. Vasanta to have another ICMR meeting where you talk in terms of ethics committees for genomic, RDNA based, protein based research to sit down and look at what the prerequisites are for people who sit on that committee. But I would definitely take that point and say yes, there is merit in the fact that you can even start looking at them at the time of patenting the products. Yeah.
Sir, as I have heard that non-biotics means now who are developing drugs, they are not developing new generation of antibiotics. They are trying to the drugs which are already in market having any effect of antibiotic property or not. And in that case, suppose metronidazole, metronidazole is antibiotic itself. Suppose omeprazole, just take it granted, omeprazole has its any antibiotic effect or not. If I want to do study, is it necessary to do the toxicological studies of omeprazole again?
If you're looking at an existing product for a different indication, okay, you can go ahead and the preclinical study has looked at some of those effects, and you can allude to that and say, based on the fact that this did show a weak antibiotic activity, I am sort of proposing a phase 1 study in humans where I am not using omeprazole as an H2 receptor blocker, but since it has similar structure, I might be looking at its antibiotic action. You will be permitted,
because
but there has to be a strong logic.
Why I am questioning about it, because as per guideline of drug development in herbal medicine, especially in Ayurveda, suppose a drug, suppose Makradhaj, suppose it is used for RTI, suppose, and nowhere it has been mentioned that it is used also for UTI or pain or anything else, suppose. And if we are trying to develop Makradhaj as a painkiller or painkiller in that way, we should go for every toxicological study. But why should we? Because of already it is in existence for 2000 years on human body.
All right.
So
if you have a strong logic,
yeah.
if you have a strong logic, you have a strong, I would say, literature review that talks in terms of, yes, the product has earlier been used for whatever property. 1. 2, initial drug screening process, and if you remember, I said we will look at a lead compound, then do what's called a pharmacological screening. And I'm sure academic pharmacologists can clearly tell you what screening is. You start looking at, A, does this drug have effect from out there here to all the way to the toe? Look at all of those systems. Have some data that yes, it does have a bit of antibiotic activity associated with a certain amount of anti-inflammatory activity. But since my literature is talking only in terms of the anti-inflammatory property, you have proceeded and developed that drug. You can always go back, allude to that mild antibiotic activity and say at higher doses perhaps, or at a modified formulation, it could be used for say antibiotic property. If the logic is strong, you can go ahead and say, I won't repeat the animal pharmacokinetics or animal studies. 1. 2, if you have a concept, you can put together a protocol called the proof of concept protocol and say, I wish to prove this concept based on my scientific rationale, which I'm sure if the drug authorities were here, they would probably take credence and say it's permitted. I've known examples where I've done it.
My point is, it should not be granted as a new chemical entity, because already it is in use. That is the basic point of mine.
I'm sure Madam wants to answer that question.
Actually, this was raised many a times, and why should you waste time in doing things which has been proven to be in use for ages together? But then, if you want global acceptance, you have to do certain things. You can confine it to Indian population itself. Nobody has any problem. But if you want to market it externally, they have their own regulations. It's not necessary that they will accept your views. Okay. The thing is, this has been discussed among experts, and initially what we had planned was that any traditional formulation for traditional use, you can straight away do human trials. That's called reverse pharmacology. Okay. But if the same traditional formulation is to be used for a new indication, then you have to do limited toxicity studies. Then came a question, for example, say something which is used for gastric problems. Now, if this is to be used for wound healing, then you don't have to go into that many tests. Okay, so you can have a local skin test or
Skin
something
allergy
like that.
test.
You have to formulate that. But I want to bring to this audience the news that, of course, when we modified the thing in consultation with the Department of Ayush, that is in your guidelines. After that, again, there was sitting for formulating guidelines for the industry, how to go about the regulatory requirements of these formulations, which have either a new indication. The active principles from these plants have to undergo all the regulatory requirements. The only question is about these traditional formulations being used for new indications.
Thank you so much.